We will post new science and technology updates here. Bioscientists use mixed reality headset, custom software to measure vegetation in the field. A lab that specializes in applying data science to the study of conservation biology has created an open-source app to harvest field data from Microsoft's. The app allows researchers to gather data as they walk and look at vegetation. Spatial data is displayed on the HoloLens screen in real time and can be stored for later analysis. You might like a major step forward for organ biofabrication. Heart disease, the leading cause of death in the U.S., is so deadly in part because the heart, unlike other organs, cannot repair itself after injury. That is why tissue engineering, ultimately including the wholesale fabrication of an entire human heart for transplant, is so important for the future of cardiac medicine. To build a human heart from the ground up, researchers need to replicate the unique structures that make up the heart. This includes recreating helical geometries, which create a twisting motion as the heart beats. It's been long theorized that this twisting motion is critical for pumping blood at high volumes, but proving that has been difficult, in part because creating hearts with different geometries and alignments has been challenging. This advancement was made possible using a new method of additive textile manufacturing, focused rotary jet spinning, FRJS, which enabled the high-throughput fabrication of helically aligned fibers with diameters ranging from several micrometers to hundreds of nanometers. Developed at SEAS by Kit Parker's Disease Biophysics group, FRJS fibers direct cell alignment, allowing for the formation of controlled tissue-engineered structures. The research is published in Science. This work is a major step forward for organ biofabrication and brings us closer to our ultimate goal of building a human heart for transplant, said Parker, the Tarr Family Professor of Bioengineering and Applied Physics at SEAS and senior author of the paper. This work has its roots in a centuries-old mystery. Over the next 3 centuries, physicians and scientists have built a more comprehensive understanding of the heart's structure, but the purpose of those spiraling muscles has remained frustratingly hard to study. Our goal was to build a model where we could test Salin's hypothesis and study the relative importance of the heart's helical structure, said John Zimmerman, a postdoctoral fellow at SEAS and co-first author of the paper. The first step of FRJS works like a cotton candy machine. A liquid polymer solution is loaded into a reservoir and pushed out through a tiny opening by centrifugal force as the device spins. As the solution leaves the reservoir, the solvent evaporates, and the polymers solidify to form fibers. Then, a focused airstream controls the orientation of the fiber as they are deposited on a collector. The team found that by angling and rotating the collector, the fibers in the stream would align and twist around the collector as it spun, mimicking the helical structure of heart muscles. The alignment of the fibers can be tuned by changing the angle of the collector. The human heart actually has multiple layers of helically aligned muscles with different angles of alignment, said Hubin Chong, a postdoctoral fellow at SEAS and co-first author of the paper. This is important when it comes to building a heart from scratch. Take collagen for instance, an extracellular matrix protein in the heart, which is also a single micron in diameter. It would take more than 100 years to 3D print every bit of collagen in the human heart at this resolution. FRJS can do it in a single day. After spinning, The ventricles were seeded with rat cardiomyocyte or human stem cell-derived cardiomyocyte cells. Within about a week, several thin layers of beating tissue covered the scaffold, with the cells following the alignment of the fibers beneath. The beating ventricles mimicked the same twisting or ringing motion present in human hearts. The researchers compared the ventricle deformation, speed of electrical signaling and ejection fraction between ventricles made from helical aligned fibers and those made from circumferentially aligned fibers. They found on every front, the helically aligned tissue outperformed the circumferentially aligned tissue. Since 2003, our group has worked to understand the structure-function relationships of the heart and how disease pathologically compromises these relationships, said Parker. In this case, we went back to address a never-tested observation about the helical structure of the laminar architecture of the heart. Fortunately, Professor Salin published a theoretical prediction more than a half-century ago, and we were able to build a new manufacturing platform that enabled us to test his hypothesis and address this centuries-old question. The team also demonstrated that the process can be scaled up to the s. It was supported in part by the Harvard Materials Research Science and Engineering Center, DMR 1,420,570, DMR 2,011,754, the National Institutes of Health with the Center for Nanoscale Systems, S10OD023519, and National Center for Advancing Translational Sciences, UH3TR000522. 1UG3HL1417801. Make a difference. Sponsored opportunity story source. Materials provided by Harvard John A. Paulson School of Engineering and Applied Sciences. Original written by Leah Burrows. Note: content may be edited for style and length. Recreating the heart's helical structure-function relationship with focused rotary jet spinning. Science, 2022. 377, 6602, 180 doi, 10. 1126, Science. ABL 6395. Cite this page. Harvard John A. Paulson School of Engineering and Applied Sciences. A major step forward for organ biofabrication. By recreating the helical structure of heart muscles, researchers improve understanding of how the heart beats. Science Daily. A major step forward for organ biofabrication. By recreating the helical structure of heart muscles, researchers improve understanding of how the heart beats. A major step forward for organ biofabrication. By recreating the helical structure of heart muscles, researchers improve understanding of how the heart beats. Science Daily. https://ift.tt.jn0w5du. Access July 8, 2022. A warming climate decreases microbial diversity. Researchers at the University of Oklahoma have found that the warming climate is decreasing microbial diversity, which is essential for soil health. Their findings are published in Nature Microbiology. Climate change is a major driver of biodiversity loss from local to global scales, which could further alter ecosystem functioning and services, Joe said. Despite the critical importance of below-ground soil biodiversity in maintaining ecosystem functions, how climate change might affect the richness and abundant distribution of soil microbial communities, bacteria, fungi, protists, was unresolved. using a long-term multi-factor experimental field C. Additionally, this is the first study documenting the differential responses of both spore and non-spore forming microbes to climate warming, and this is the first study documenting the predominant role of warming in regulating microbial biodiversity. Thanks for watching. Please subscribe my channel for more videos.